Yes. Okay. Hello, everyone. I'm Michal Mielcarek. I'm working at the Imperial College London, and the talk will be about the skeletal muscle pathology in HD with the implication for therapeutic approaches. So basically, to add to another question to Daniel's talk is where to apply any therapy in Huntington patients. Just briefly, as Daniel mentioned, Huntington disease is inherited disorder caused by CEG expansion, and all of us, we have all the CEG repeats, and we are unaffected when we have below 35. Then 36 to 39, we have so-called incompleted penetrance, and over 40, we basically will develop the Huntington disease, and there is also a juvenile form with over 70 CEG repeats, which manifest very early in age. So what we know about the Huntington gene was cloned in 1993. However, we still don't know what it does. And we know that it is expressed in all tissues and is also expressed through the development. And we know from the mouse studies that if you knock it out completely, the embryos die at day 8.5. So the Huntington gene is a large protein. It has 3 or 4 hit domains, and then we have also this Polyq Track, and with the followed by proline-rich domain. So it is, as I mentioned, just that is expressed everywhere, and the CEG expansion, which is translated to the glutamine. So we have the Polyq Track makes this protein prone to aggregate, and we can, at least in all the mouse models, we can detect the aggregates of mutant Huntington throughout in different cells and tissues. In the lab, what we can study, we have 2 types of Huntington disease mouse models. The first are the R6 models, so this is only a fragment of the human exon 1 under the Huntington human promoter with expansion from 130 to 200 CEG. Keep in mind that the mice only have 7 CEG repeats, and so to induce the HD phenotype in mice, we need to go over at least 100 CEG. And then we have a so-called full-length mouse models when we introduce the mutation into the mouse genome. So the fragments models, one might call even overexpression models. This is like the full-length models are replicating the human situation when we have a mutation within one allele, while the second one, at least in the heterozygote, is not affected. And just to compare the phenotype, we brief the fragment models. They are very quick to develop all neurological and molecular symptoms, and they die within 3 to 8 months of age, while the full-length models are representing more the adult onset of HD, and the first symptoms can be detected around 1 year of age. And just to briefly mention that there is also people are mentioning that the fragment models are basically a representation of juvenile form of Huntington disease. They are highly comparable, so any phenotypes found in the fragment models like R6 models, and then the full length like HD, HQ150 can be detected in both. So the weight loss, neurological symptoms aggregate profile, muscle pathology, cardiac dysfunction, and glucose homeostasis. And this what, as I mentioned before, the Huntington is ubiquitously expressed, and we found, at least in mouse models, that the aggregates, the toxic aggregates, accumulate throughout the tissues, and we can also think that this might happen the same in the humans. So there is an obvious skeletal muscle atrophy or dystonia. There are problems with the cardiovascular system. There are also problems with the digestive tract and with the thyroid. So the main hypothesis behind this talk is that if we have a pathogenic stimuli that affects the CNS within the time, that will also affect the skeletal muscle, and then basically as a negative feedback loop, we will affect the CNS. So by improving skeletal muscle function, we might also delay some symptoms and keep the CNS more healthy than without the intervention. So in our previous studies, we found, so this is all the data based on the mouse models. We found that indeed the skeletal muscle weight is affected within the time. This is different types of muscles like quadriceps, gastrocnemius, tibialis, EDL, and soleus. So a representation of slow and fast and mixture of both types. This is in R62 model, and this is in the full-length HD HQ150. So you can see that there is ongoing skeletal muscle atrophy just based on the weight of the muscle. And then this is accumulation of aggregates in all type muscles in both models. So within the age between 12 to 14 in the short R62 mouse model, we see a increase of aggregate load, and this is at 22 months in the HDHQ150 model when the aggregates are apparent. So next we studied what does it mean for the muscle. So we use an in vivo equipment to measure the physiology of HD muscle in fragment model. So we basically innervated, we use a detector. We use first an impulse to deliver the impulse to the mouse, and then the detector to measure the muscle force, the tetanic tension, the muscle twitch, the time it takes to twitch, and then the activity of motor units. And this was done all in a living animal, which was fully anesthetized in vivo. So we provided the impulse through the sciatic nerve, and then there was the detector was attached to the tendon of the tibialis or EDL muscle. So we found that the maximum twitch was significantly reduced, both in EDL and in TA muscle, and the maximum tetanic tension was also significantly reduced, which basically was somehow expected based on the muscle weight loss. In addition, we also found that the neuromuscular function is also affected in this model. So by scoring the motor units, we found that there was a progression in decline of active motor units. We are not sure whether the motor neurons are dying or they are less basically functional. And we found that this by scanning a number of molecular transcripts, that there is an ongoing switch from fast to slow type muscles. We also were interested to look into the purine homeostasis and energy metabolism, and we found that there is a repetitive pattern between fragment and and full-length models indicating that the purine homeostasis and energy imbalance is also affected in HD muscles. Then we developed also a cellular model of skeletal muscle pathology in HD, and we found that by MTT assay here, that when the mutant Huntington is expressed, there is a high prevalence of cell death, and also we measured the mitochondria volume, which are enlarged, defragmented based on the volume and the maximum ferrite diameter. So the question is all the pathological changes coming after the CNS starts to degrade or degenerate, or there is also an intrinsic element like the mutation within the skeletal muscle that leads to HD-related skeletal muscle atrophy? This is still open question. We don't have a proof or disproof that both pathways are not active. However, we did recently a study by applying a activin receptor, which is the agonist of myostatin, and one you might know that myostatin is a negative regulator of muscle mass. So if you knock down normal myostatin in animals, you will increase the muscle mass and muscle function, likely leading to muscle hypertrophy. So by applying the activin receptor and cutting out or depleting the myostatin function in vivo, we found that we were able to restore the motor units almost to the wild type level. And we found that this is a typical test for the mouse, which basically don't show the tremor up after treating with the activin receptor 2B, so by depleting the myostatin function. Moreover, we were of course able to restore the muscle function to the Y-type levels, and this was ongoing with the ongoing CNS degeneration, because you have to remember that the activin receptor didn't penetrate the brain-blood barrier, so there was no effect on the CNS. It is a pure effect on the skeletal muscle. And finally, we were able to extend by around 25% the lifespan of the R62 mice. Thank you for your attention. So any question, then I'm happy to answer.
Yeah, thank you so much, Dr. Michael, for your precious words. Thank you for presenting among us. Dr. Michael, we are going to have a question and answers session at the end of the conference.
Okay, yep. Yeah. Happy to answer any questions. In any event, you will have my email. I am also happy to answer, outside of the conference if somebody has a more specific one.
Thank you. That's so kind of you, doctor. Thank you so much.
Thank you. Bye, everyone.